introduction for the next speaker, which is Marisa Papaluca and she's talking exactly, dispelling the myth, looking at benefits and risks. So thank you very much, and the floor is yours.
So thank you very much. I hope I don't annoy you too much with my presentation after such brilliant speakers. Both Elise and Robert are so eloquent. I'm really impressed. First of all, I want to thank Clinam for inviting me to give a perspective which is completely on the clinical side. So you will have a sort of different kind of perception on what nano-based medicinal products and devices can provide. I also want to pick up on 2 points, very important from the speech we just heard. The first one, are we using enough, exploiting enough academic research? Recently at a conference in London, I just made this point, that if we want to have success in the clinical endeavors, we have to ensure that we not invent the wheel all the time, and that the academic research actually blends into development along with manufacturers. Because I see, as an independent advisor at the moment, so much disconnect. I work at Imperial College London, where we have a fantastic academic research. And then in my private consultation, I see companies reinventing the wheel. So where is all this waste? This is not acceptable. I think we have to reconfigure how health products are manufactured and reach the patient. The second point is that from what Robert just said, I'm very pleased to say that indeed, at least in the European system, we have learned society and groups which are actually providing tools to regulators rather than just criticizing regulators. And this is absolutely laudable. Although it's not always transparent in the actual assessment report which kind of tests, because these are most of the time proprietary. So I would like to dispel the myth, because just today someone told me, oh, nanomedicine term may have a negative configuration because people feel that nano can be dangerous. So I want to dispel the myth, saying how the risks, how the problem are looked from a clinical standpoint. So, of course, the views I'm presenting are mine. It's not Imperial, it's not the European Medicines Agency, and where I worked for 25 years as a senior scientist. So, let's see the reality, independently from the health system. Aerospace industry, huge, is where the power of nanotechnology found its own initial house. In the creation of the screen for solar energy, they were all based on nanotechnology. It was fantastic. Then look at the dyes, the textiles. I'm in white, but I'm not sure whether I'm without nanoparticles on my attire. The cleaning products. Actually, the cleaning products are the one who initially created so many difficulties because the inhalation of nanoparticle for glass cleaning. Cosmetics. Now we know, in Europe at least, but at the worldwide level, the presence of nanoparticles is to be declared and is to be justified. Food wrapping. We had the major activities with the EFSA when I was at the MEA to see how the food wrapping could be involved in the absorption across the food of nanoparticles and what consequences would be there. And finally, we have health products. I want to bring this one as an important slide because Wim de Jong has been the guy which, since 20 years, has been really heralding the need of a proper risk assessment. And already at that time, this is a slide he presented in the conference at the AIM in 2010, he says, it's not just the pure nanomaterial, it's the convergence of nano approach in many different aspects. So you have biological sciences, which actually entirely, especially nowadays, work at the nanomaterial, at the nano size interactions, and all the rest you can see in these slides. What is the message here? Nanotechnology is not just one product. You find the nanotechnology in your chips for your computer, in your lab tests, etc. So it's really very appropriate what was said by Elise, that maybe the revolution actually is already unfolding. we just into it, so maybe we don't see it properly, but it's already with us, okay? So therefore, it's very important to think about the risk associated with nanoparticles. I apologize if it's too elementary here, because here I heard a presentation today of outstanding science. This is very basic, okay? So absolutely appropriate to say which level of risk to living organisms and to the environment is acceptable, what we are prepared to accept. This is a question which is not very frequently posed, because every time you go to a conference, you have a particular angle. So the level of risk I can accept for a medicinal product for a particular condition will be very different from the level of risk I would accept for the dye on my textile. So this is a question which would require much more work, how we define the level of acceptability for a risk. And of course, along with it, once you have the response to that, you have to have a strategy for assessing the risk and also for mitigating the risks. And this comes with all the beautiful information we had just provided by Robert. But again, do we need, can we legislate before having an understanding of where we stand? This is a question I cannot answer, because I'm very much restrained to my own little planet of medicinal products and devices. So, but let's think again. What is a risk? This is from Wim. Very, very simple. It's a combination of the likelihood of occurrence and occurrence of an harm and the severity of this harm. So this is a risk. And then you have to have a margin of safety. So how you can manipulate the levels of exposure depending on the dose and the no effect level. So for the researchers in animal studies, for example, essential. I don't see this so much. There is sometimes just very little on the margins of safety in animal experiment, where everything, especially when something is particularly effective. Of course, if you have no exposure, there is no risk. So risk is a possibility and not an absolute value, okay? So, and also another element which is very important, particularly when we speak about nanotechnology in health products, is linked to interaction between that particular type of nanotechnology-based product and the receiving organism, okay? And the exposed organism, be an animal or a human. So then what is a nanotechnology-based product or medicine? The one who have been with me long enough will see a change in terminology. What you see here is a working definition we elaborated at the agency, I think it was 2009, something like that. And the title of this, the definition of a nanomedicine, because the agency at that time was only mandated to speak about medicinal product. So it was extremely sensitive to go outside the border of medicinal product. But if you go through this one, you see that what was the definition is a design system. For the first time, we spoke about a system, a molecular system, and not about a new active substance or a new substance. So it's a system. And I've seen even in this conference, magnificent example of that. Then it says at least one component should be at nanoscale size. So not the entire system should be nano, but at least one component of it. And then this design and the system should have specific and definable properties and characteristics. So those will have to be related to the specific application. And again, I wish to thank the group who work on Refine and what has been presented, because absolutely crucial that you adapt the kind of test and definition, and depending on which specific nanotechnology you apply. But also, the design is associated with an expected clinical advantage. So here you see that even in the definition, here we have the benefit and risk. I will demonstrate that this definition is over now. The concepts are still valid. The definition does not apply just to medicinal product. And we will see how the component of the nanoscale size is not what was taught at the beginning. We were thinking at the beginning at the size of a liposome, at the size of a nanoparticle. No. It's a specific component, not all of it. So this, I think, introduced the concept that, by definition, a clinical, a nanotechnology-based product for health, by definition and by design, should anticipate some clinical advantages. So what is special about nanomedicines? You know this much better than me. Variety. I will not go through all of this, but just to demonstrate for those who have been at this fantastic conference how behind we are. In this conference, with the exception of the molecular nanorobots, which are completely new, I've not seen here around, but we have the click system, we have a wealth of new ways of exploiting nanotechnology for new clinical applications. So we have a variety of sizes and structures, but even within the same class, I would say within the iron nanoparticles, within the liposomal formulation, we have quite a variety. And what I wanted to point out, I don't know whether there is a pointer here, no, maybe not. What I want to point out is not just which kind of sugar you have around the iron nanoparticle. I want to point out a little bit the fact that every single of this product has been developed for a very specific population. Because again, as I said at the very beginning, the risk is the interaction between the substance and the specific recipients. So you see in these slides that there have been different choices of the various components. Oh my God, this is also late. Anyway, you can see that the concept is the variety even within the same class. And the clinical range. We go from cancer to infectious disease to pain. This has been largely demonstrated by a number of presentations in this conference. I will not dwell too much on that. I want to go specifically with some example, starting from a long time ago and coming more recent examples. So I will not discuss the structure of Calyx. I just want to attract your attention on why Calyx is on the market and under which condition. As you can see, it was in Kaposi, AIDS-related Kaposi sarcoma, the pegylated liposomal formulation was more of the substance was more effective than either a combination of bleomycin and vincristine or a standard doxorubicin. So because of better efficacy, okay? So the benefit risk, the risks are related to the doxorubicin or well known, but the benefits are really outweighing the risk in terms of more effectiveness. But if you look at the metastatic breast cancer, it's not more efficacious, but here you have better results when it comes to cardiac complication. So this is, I think, shows you that the same structure, depending on the recipient, you may have different weighing of benefits and risk. I have still 2 minutes, so I will go quickly. This has MEPACT. This is a multilamellar liposomal formulation. Again, this is the study being conducted in people between 2 and 30 years of age. And I want you just to point out the duration of the follow-up in years. Okay, if you have someone who is 30 years old, 20 years old, 2 years old, have an extension of the overall survival for 12 years, is absolutely unobtainable. Was absolutely unobtainable before this was coming across. It's used not on its own. It's just not a lovely nanotechnology-based multilamellar liposome, but is used in conjunction with chemotherapy. But the results are clear. As you can see at the very beginning, this is a Kaplan-Meier curve, the opening and the advantage for the survival is visible at the very beginning of the curve. And this is again, another liposome. Here we have a sirolimus, a nanocrystal dispersion. And again, this is advised to be used in conjunction with other immunosuppressants to prevent kidney rejection. But look at the separation of the line. The black is all patient, but if you look at the one with the association of cyclosporine, and then after a certain period, just with the rapamune, shows that this particular product and also other example have a flexibility on how you can introduce those in clinical care. So they're not an absolute, but they can become an absolute depending on the stage and when you use them. This is even more impressive. This is the treatment of neuropathic condition linked to a genetic defect whereby amyloidosis accumulates in the nerves. And if you look here again, the response, 18 months, is absolutely extraordinary. The blue line is patient treated with this double-strand small interfering RNA. This is interesting, it's 2018, it was before COVID. And this is one of the very first successful genomic medicine products, because this is actually interfering, reducing the amount of the misfolded protein, which creates the accumulation of amyloidosis on the nerves. This one is Lutathera, which we like to call it the very first nanotheranostic. This is for a very rare condition, which, as you can see from the graph, has quite a big toll in very few months on the population. The beauty of this is that you have targeted with a peptide to particular type of tumor which overexpress statin receptors. The side effects are terrible, of course, very severe, because also a beta emission, because the radiation associated with lutetium. But it's interesting that the product has been formulated and manipulated in a way that the penetration of the beta emitting is 2 nanometers. So in principle, should be very strong on the cell where it binds, but quite limited. But of course, being a radiopharmaceuticals, there are lots of risks, which we know very severe. Oh, my speaking time expired. This is the last one. This is the thing which shows that our definition is obsolete. Unless we interpret that at least one part is a nano-sized. This is a depot foam particle, which has been, which is an interesting application of nanotechnology-based product, because you have a treatment of severe pain, which in the past was linked to bupivacaine plus opioids. With this product, you have a reduction of an opioids treatment, which is terrible. So what is special then about nanomedicines? Again, if I was to go back, I didn't send the slide before, I would say nanotechnology-based medicine, because nanomedicine is not really fully applicable anymore, because we move away from just the size of the product. We just look at the various elements of the system. Like I showed in the one before, you have the vesicles are less than 2 or 3 nanometers, I think. Then they enable novel mechanism of action. I mentioned very briefly the genomic medicine. They combine multiple functions. This is the case of Lutathera, for example, for precision medicines. They allowed, and the one I just showed, fantastic repurposing of existing active substances. And also, if you go into the site of the European Medicines Agency, you will see how active is the post-approval life of these products. They have continuous update, both on the quality and manufacturing and on the clinical use. Takeaway message. We had, for many decades, people were considering science fiction, so it was not really a real thing, it's just a scientist fantasy. Now is a reality and benefit patients in many ways. The other thing is no risk is acceptable in absence, if there is no evidence of benefit. So as regulators, we want to have the benefit, and whatever the risk, then we will make the benefit risk. But in order to weigh the risk properly, we have to have a full benefit assessment. And so thanks for your attention. And we can discuss, because there is a lot to discuss. Thank you.
Thank you very much for this very but inspiring talk. Because of the very interesting talks, we have time for one question, because otherwise the exosome people are coming to kick us from. So, other questions?
Hello, thank you. So for the panelists, one of the safety concerns that we have been seeing this event is immunogenicity. My question is about different exposures from different products. We might be seeing the same component in different products, different vaccines, and different any kind of product. So our framework for safety evaluation should change in this direction on considering different products?
Yeah. Yeah. See, I should have done like the gentleman yesterday and started with the conclusions instead of the beginning. Thank you for looking at my another day's slides. I believe, to be very brief, because other people are waiting their turn, I believe that the traditional model we have for analyzing this, whether the common law, the civil law, or the existing frameworks, is inadequate for exactly the reason you said. You will have the same paradigm for exposure in many sources, and also you will have many different exposures that interact with that can't easily be traced. Simple example, you have a medicine that is purchased in a pharmacy, and you could use the credit card to find out who bought it, or the insurance information to find out who bought it. That doesn't mean they used it, and it doesn't mean they didn't give it to somebody else. Oh, you're feeling sick, take it. So you can't precisely look at exposure the way we are accustomed to on the bulk level. And therefore, we need a new model. Because if you don't use the new model, you have a traditional model that looks at a harm and goes back and drags in everyone along that way. And it's going to be unfair for two reasons. It's going to drag in people who didn't do anything to be part of that path of analysis, and it's going to make the true reason for that harm extremely elusive and always in doubt, and always much more subjective than it ever was. So I believe we will have a new model for tort liability and for other kinds of liability and responsibility. And I invite you as a scientist to be part of that by putting in your input of what that should look like.
And if I can add a little things, this is a deja vu for me, because I was there at the very beginning of human recombinant DNA-based products, which was another revolution more than 20 years ago. And the point is that, at least in the European system, we have a system of pooling expertise all together. So if there are, let's say, the same monoclonal antibody, and we see that there is a neutralizing response from the recipient, and then the same the same monoclonal antibody targeting the cell comes in another product based for another indication, this knowledge of what happened with the first one is not lost, because we have a collegial kind of approach and a peer review approach for every single thing. At a certain point in time, it will become standard, like for the CHO produce human recombinant proteins. We knew that even in small quantity, residual cell line proteins could cause a very severe allergic reaction. So there was a specific warning for that, and this was taken into account. Yes, your question is very well posed. It will take time and experience from the regulatory scientist to identify exactly which one would trigger a cross-product negative response. And did I answer your question? Okay, thank you.
So I think we thank the speaker again. All other questions can be discussed over coffee. Okay. Thank you very much.